Champion's preclinical platform is based on biomarker tomographs. Biomarker tomographs are a better mouse model for evaluating drug candidates, oncology drug candidates. So biomarker tomographs are derived from the direct implantation of human tumors into immune-deficient mice. The tumors are then grown and propagated into subsequent generations of mice as whole tissue fragments, so that the structure of the tumor remains intact, and we use various proprietary methodologies to ensure that the basic biological characteristics of the tumor are maintained. The objective is to ensure that the tumor that is growing in the mice correlates very closely to the tumor that was removed from the patient. This enables us to test drugs against the mice and evaluate how the tumor responds, and then predict how the patient then will respond to the same drugs. We leverage that methodology for our personalized oncology patients to evaluate dozens of different drug candidates in order to evaluate what may be the best treatment option for the patient. And we also leverage this as a platform to evaluate drugs for our own pipeline and for the pipeline of other pharmaceutical and biotechnology companies. It's well known that traditional mouse models and the preclinical testing models that have been used for decades are not predictive of what will occur in the clinic. In other words, drugs that successfully navigate through preclinical trials only have about an 8% success rate when they enter into human clinical trials. That leads to a tremendous expenditure of money and resources towards clinical trials that ultimately fail, which of course also results in clinical trial patients that receive treatment that has many negative side effects and very little efficacy. We believe that our technology offers the ability to shift the paradigm towards expending more in preclinical testing, because this is a more expensive process, but enables us to predict exactly what will occur in the clinic, and therefore eliminate drugs that do not have potential to proceed successfully through the clinic, and predict what is the best application of the drugs that do succeed in the biomarker tomograph testing and how they should be applied in the clinic. This offers the ability for us to dramatically increase the rate of success in the clinic, and what we expect it to lead to is for companies like ourselves and other small and large pharmaceutical and biotechnology companies to invest more in preclinical testing and to save money in clinical testing by bringing more efficacious drugs into the clinic, which ultimately is good for companies, it's good for the drug development pathway, and it is ultimately very good for patients.